thank you for the organizers, for Peijion and Peter for inviting me here. It's always great to come back to where actually my cryo-EM experience started. So I'm very happy to be here, and also an opportunity to catch up with some friends I haven't seen for a while. Yes, so I think we are in a way continuing the same theme, because I was also trained in the tomography and doing a lot of cryo-electron tomography, and facing a similar problem that Martin just mentioned, that we see so many things, and this is why we like tomography, but very often we don't reach the resolution that we need. And so we had to also be creative about how to identify some of the proteins that we see in our cell. So just a bit of background kind of to understand. So in my group, we are interested in this magic process of fertilization. Yeah, this is the start of all of us. And to be honest, I think this process and the gametes that are making this process, there were a good sample from the early age of EM. And if you go back and you remember when was the electron microscope was discovered, you can see that already in the 50s, actually, there were already a few images coming from sperm cells. So what you can see here is the nucleus, the head of a sperm cell. In the center, you see really beautiful parts of the tail. And the center is the axoneme. This is the motor apparatus that we'll talk more during this talk. And what you can see also on the most right are some ion channels on the membrane. So all of these were already found or discovered so many years ago. But again, the limitation of EM is that we don't know what are they. So we had to work quite a bit to reveal these proteins. So these are some classical EM, but of course there was also cryo-EM done on sperm as well. So the whole cryo-EM, cryo-electron tomography came along, and the group of Daniela Nicastro was kind of leading, studying sea urchin sperm as the model species. But from all this kind of all the classical EM data, it was very clear that mammalian sperm are much more complex. And this is kind of the challenge I took upon in my group. And I like this drawing very much because it is based on all this, all the EM data. And what you can see here, that yes, you have the head where you will have the nucleus, and the major part of the tail is the motor apparatus, the axoneme, but around it in different parts of the cells, you will have different structure around it. So in this area that is called the midpiece, you will have the mitochondria arranged around this axoneme. So it forms this helix, and this is a kind of shown in more detail here. So you have in the center the axoneme, but you have some outer dense fibers that are connecting to the axoneme, and around all of this, you have the mitochondria. And if you move on to further away from the head, so to the area that is called the principal piece, so you don't have mitochondria anymore, but this structure that is called the fibrous sheath is now surrounding. So it's all much more complex than in a non-mammalian species. And then working in sperm brings its own challenges. Because these cells are transcriptionally and translationally silent, so no ribosomes in these cells to average. And so there isn't a good way to culture or overexpress. So many of the genetical tools that are available if you work on a cell culture are not available for us, okay? And also, this is probably one of the cell type that is the most diverse. So what we learn on one species not necessarily is relevant for another species, even though maybe the overall structure seems to be the same. You have head and tail, but I will show you that on the molecular level, it is so much more complex, okay? Plus, to make life even more complicated, many of the proteins are actually tested specific. So it can be one protein that is you can find in general proteomics, but then in sperm, it will be a very specific isoform. So with that in mind, we went on to be able to still develop tools to study mammalian sperm. And this is kind of our maybe overall simple workflow. So we get the sperm from either different species or human. We just put it on EM grid and we do plant freezing. We can do some fluorescent imaging if there is a marker that can work. And then it's either we go directly to a transmission electron microscope or we go through the field milling if we want to look at parts of the cell that are a bit thicker. So from the beginning, we kind of took, in a way, a holistic approach where we analyzed what I will call from head to tail, the whole cell, because that is one of the power of electron tomography, because once you have your sample in the microscope, you should, I think, look at everywhere. And we try to average whatever possible, I think, by now in the cell. So some examples that you can see here. Some of them are related to the microtubule structure. So in the center, you will see the axoneme. So this is the doublet of from the axoneme, and this is a central pair. These are in the tips, you find only microtubules, and this is this average here, but also the centrioles. But again, we run into this problem that many of these proteins were not identified before. So one of the approach we took was to combine it also with proteomics, but more specifically cross-linking mass spectrometry. And that worked well for one example, when we analyzed a protein that is on the surface of the mitochondria. But maybe one of the limitations of cross-linked mass spectroscopy, and this is also something we did on the whole cell, so we did not isolate anything. So the cross-linking mass spectrometry was done on the whole cell, so you end up with a long list. So it's never only one protein that is really interacting with your protein of interest. So we kind of hit a bit a wall there, moving on to analyzing in more detail the major motor apparatus, which is the axoneme. That just show you again this scheme. So now we are going to focus on this part and specifically on the doublets from this part. And then, so to come up, to go about trying to identify the proteins that are in this assembly, we had to be more creative, but the first idea came from something we tried to avoid for a long time. So this is, I like this because it's one of our first EM session on the pig sperm, and it's made of this high magnification projection images. And if I zoom in only in the end piece that you can see here, what happened is that we lost the membrane, and then you see the microtubules are actually spreading a bit out. So we spent about 3 years trying to find the right condition, how to plunge sperm so we do not lose the membrane, because we wanted to do a tomography of the whole cell. And the center point, we decided we do not continue to fight this, even though we did find ways how to keep the membrane, but we take it into our advantage, because now the microtubules, when they are spread, then you can treat them as single particle analysis workflow. And this is what you can see here. How does it look? So you have really nicely microtubules spread. Of course, this is not normal single particle data collection, because we do not have the sample everywhere, because the whole cell is still there. We did not isolate anything, yeah. But we got to a very nice resolution that we could look at the microtubules, and from our tomography data, we saw that there is a protein that binds inside the microtubule. So you can see it here, and we got to a resolution that we could identify this protein, and we validated also with our cross-linking mass spectrometry. So this is a SPACR9 that you can see here. And because we got such a nice resolution, we actually resolved another microtubule inner protein that it's a bit harder to see, because it's a very thin filament that binds along the microtubule protofilament, and this is SAXO1. So maybe this was kind of the easiest part, because we worked initially only on these end pieces that have only these singlet microtubules. But of course, the challenge was to go and to do it to the bigger assembly, which is the major part, which is this axoneme. So just a reminder that you have a central pair in the middle and you have the doublets around it. So this is a cross section you can see here. Again, you have, this is in the whole cell context. So you have the plasma membrane and you have this fibrous sheath that makes, again, the whole assembly thicker. This is actually tomogram, so I can play it. But what's nice is that you can already see that there is a repeating units of proteins along this structure. Yeah, so you can see this the whole tomogram, and we did do the averaging for this part, for this doublet microtubule, and this is an example where our comparative studies were becoming interesting because what we found is that different species have different amounts of, what did I do? I don't know. I don't think I clicked anything. Okay, gone. So we saw that there is a slightly a variation in terms of the microtubule inner proteins that are inside this microtubules doublets. But of course, as I said, what we wanted is to identify those proteins. So for that, we needed to be a bit more creative, or in a kind of more correctly, the postdoc in the group, Miguel, was digging out into kind of all the literature and found that there is a way to put, to kind of gently disintegrate the whole axoneme structure. And this is kind of the workflow, how we do that. So what you do is we gently remove the membrane by adding a detergent. And then we add DTT to remove this layer of mitochondria around, so in this area. And then once you add ATP, the microtubules, the dynein start to work on each other, and the whole structure start to disassemble, but in a very gentle way. And this is how now our sperm look like on the EM grid. So it doesn't look like a sperm so much anymore, more like an octopus, I think. But if you look in a more higher resolution, you see the microtubule here, and they actually even return this outer dense fiber that is still connected to the microtubule doublet. So that's in terms of the sample preparation. And then we went through for a single particle analysis, a complete processing approach. So started with just the doublets, but then zooming in on the sub structures, and you can see that we reach pretty good resolution in all parts of this structure, to the point that we could really identify these proteins. And then what was really nice, that we did have our subtomogram average from before, and when you compare it, you see that we did not lose many or at all proteins in this process of this disassembly. So this is a kind of the whole overall structure. So we identified more than 65 proteins, which is quite a bit. And now what you can see here is that every protein other than the microtubule is colored in different color. And you can see on the right some examples of how the quality of the map that allow us to identify those proteins. And this is what you see here on the bottom is kind of a cross section. But what I wanted to highlight in this structure that we have, it is quite a gigantic structure with more than or close to 1.5 million 9 hydrogen atoms, which means it comes actually from more than 180,000 residues. And just to deposit the structure was quite a struggle. So I'll be happy to talk with the EMDB people to make our life a bit easier in the future, because it's, as I show you, it is just getting bigger, but that's, we can do it later. Yeah, but I think the best is actually to show this structure when kind of in the movie. So what you see now is the map. And what we will do is we'll color the map based on the different protein. White stays the microtubules. And now we color, and this is the model that we built in. And And if we turn off the microtubules, that you will see that how all these proteins are interconnected together to form this huge structure. And then very interestingly, we see crosstalk between the inside and outside of the microtubule. We focused mainly on the inside of this structure. And obviously, when you have a structure of this size, there is so much to learn from it. But I will spare you all the detail. I wanted to give like one example that became a very interesting case and related to this diversity in general in sperm. So in parallel to our work, the group of Ray Zhang in Washington University, they determined the structure of the doublets from in high resolution from sea urchin sperm. And then we talked and we started to compare our structure. And this is where it's becoming interesting, because sea urchin sperm, sea urchin are what we call external fertilizers, and mammalian species are internal fertilizers. So there are different media that they need to operate. And so on the ultrastructural level, there are differences, because I told you that we have the mitochondrial sheath and we have the outer dense fibers, and sea urchin sperm doesn't have it. But when you zoom in and you look inside the microtubule doublets, you can see that mammalian sperm has substantially more of this microtubular inner protein compared to sea urchin sperm. And we also compare it to cilia from other species, so from trachea, and we can see that really the case of mammalian sperm is the one that has the substantially more microtubular inner protein. So the A tubule is almost fully packed with the proteins. And then from this analysis, we could see and analyze kind of more a bit of the evolution, perhaps, because we compared also to doublets from chlamydomonas. And specifically, we know that what we found is that the A tubule is full of this protein that is called tectin that forms filaments. And then if you go kind of from the more simple one, the tectins are actually found in the B tubule. And when you move to sea urchin sperm, so that you have only 3 tectins in the A tubule, and then cilia from the trachea has now these 4 tectins, and when you move to sperm, has completely full A tubule. And so I have also a short movie that summarizes it, because it's also very interesting on the structural level, because what we find is that the expansion of these extra microtubular inner proteins that is done by one version of the tectin that is called tectin 5. So here what you see is all the tectins together. But what you can see is that the basic ones that's also found in trachea is 1 to 4, and the expansion is done by only these 5 tectins. But even though it's the same protein, the same that is expressed, it has different conformation. So A, B, and C form this continuous filament that you can see here. But then if we look to the D and E form, so then they form a discontinued filament. And the reason is that because they accommodate another protein that is called NME7, and this now, if you look at the details, you see it's discontinued. One of the tectins, we find only 1 copy in the 48 nanometer repeat, and then another copy we found that doesn't form any filament at all, but just bind diagonally. And this is all the same protein. So it's very interesting. I don't think it's something that it's very easy to predict, that you will have one protein that will bind in a completely different ways in the same assembly. Yes, so I think what's I found it's very useful, or what you can do now with a structure like this is that what we did, we tried to map in all the known knowledge we have on cases of infertility. So this is what you see here. So all the colored one are cases that proteins that we found were already associated with infertility. But what you can see is that a lot of these other proteins, all the gray one, they were not recognized yet. as part of the assembly. So we feel that we are providing to the community a very good resource, because we tell them, so this protein, this is where they are, and this is what is their interaction networks. And that's something that is very important for the fertility field. But so all of this was published already last year. So what have we done in the past year? Then we moved on to, as I say, go bigger. So now we analyze and average the 96 nanometer repeat. And the way to go there is because now it's analyzing the proteins which are on the outside, because what I've shown you so far, it's only in the inside, because they repeat every 48 nanometer along the axoneme, but now this is the 96 nanometer. I don't have the time to talk about this structure, really super fascinating structure as well, but just to give you the number and why we do need to talk with the EMDB, because the number of proteins we have now is 181, and then our structure has now more than 4 million atoms, and residues more than 500,000. So it's just getting, bigger and bigger, which I think it's great. This is how it should be. I think that is one of the strengths of cryo-electron tomography. We are not necessarily limited by size, but just kind of to summarize, I want to say that I see it's really happy days in cryo-EM, because specifically for the field that I'm working for, the fact that we can do de novo protein identification is amazing, because we don't have so many other tools. I'm very happy that we do more and more single particle analysis in a more native setup. And obviously this is not the only example. Nowadays there are more and more examples coming out. And to finish up, I said that I'm happy, but I have some challenges moving on to these bigger assemblies. I think we discussed briefly in the morning, earlier morning session, that we need to improve our model building tools and refinement. For example, this 96 nanometer repeat structure, we cannot refine as a whole. We had to break it to 2 parts, and then each part takes 3 to 4 days to do the refinements. And it is, even the to cutting is not straightforward, how you divide and put them. And data deposition become a big issue for us. I think a discussion that the community should have is about the whole this composite map, the position compared to consensus map. All of this I think we should discuss, because, for example, for this 96 nanometer, we're talking about more than 100 maps, right? So you really want us to deposit all of this? Is it useful? But I think also falling on what can the community get, because even downloading a coordinate map of these 4 million atoms, maybe structural biologists know how to deal with it, but it's not very accessible, I think, people outside the field. And that's also something we should think about. What do we do if we go bigger and bigger? And so on these notes, I will finish. I would like to thank my wonderful group. Most of the data I show you, I mentioned, was from Miguel. And we have excellent support in our EM Center. I've been really fortunate to have good collaborators from the veterinary faculty that provide us all these sperm from strange species. And lastly, the funding. And thank you for your attention.